Welcome, everybody, to the initial edition of the DMT Quest podcast. My name is John Chavez, founder of the DMT Quest movement. I'm here with the one and only Dr. John Dean, featured in the DMT Quest documentary. If you haven't watched it, you better go check it out. He published one of the most interesting and important papers of 2019, and maybe even to date. So I'm just happy to be involved with this young scientist that's looking to change the world. Thanks for joining me, Dr. Dean.
Please call me Dr. John.
I will, man. Well, we got into a little bit of your background and how you got into this endogenous DMT research in the documentary, but I want you to go into a little bit more detail so the public understands exactly where you're coming from and what your journey was onto this interesting molecule research.
Yeah, so got my start in academia for my undergrad. Wasn't really quite sure what I was going to get into. I was playing in bands a lot in those days and considered maybe majoring in music, but it just wasn't classical guitar or anything like that. Wasn't really my style with that stuff. So yeah, I always was into science. So I kind of started taking some science courses. And so to be candid, then I had a close friend of mine just kind of die unexpectedly. He was also my age. This was I was 19. And yeah, it just sent me on what is it? If I'm lucky, quarter life crisis, right? And yeah, things got existential pretty fast. And
Did,
so yeah,
did that kind of spur you into start thinking about things in, I wouldn't say a spiritual fashion, but just like bigger questions about life?
yeah, totally. And what really took hold of me there was, so I did kind of do the Western medicine thing, and then was prescribed the usual benzos and whatnot, which helped obviously to calm everything down, but then sort of made me like an emotional zombie. But nonetheless, it was what really spurred my interest in neuroscience, because the idea that just like this tiny little pill molecule could change my brain and change my reality so much. So naturally that got me thinking of even things like alcohol, caffeine, things that are already really readily acceptable in culture. Uh, the same idea that just this little molecule can interact in your brain and change things. And then of course there was psychedelics, right, which are the extreme end of that spectrum. So all that really got me into neuroscience. So I started taking a bunch of psychology, like neuropsych classes, because I was very interested in the mind matter sort of dichotomy. And kind of got through all the biology rooted psychology courses, and then realized that I would be better as like a bio major, and then ran into the chemistry classes and loved that. And then just changed it to chemistry major. Took 6 years off or so to play music, and then came back and finished. But it was refreshing, because when I came back is really when some of the seminal studies kind of came out with psychedelics and neuroscience, like the 2012 fMRI psilocybin Carter Harris study. So when I came back to finish up my undergrad, it was really a great time and the ideal time to get back into neuroscience, because those were my interests ultimately. And so I finished the undergrad, and then yeah, it was really, there was no debating. I knew I wanted to pursue at least a master's, or even a PhD. And but I also knew that I wanted to do something in neuroscience and get out of just like straight chemistry. So it was around that time, that I reached out to Rick Strassman, and just told him of my interest in endogenous DMT, because all throughout that time, the existential time with my friend dying and everything, that's right around the time that I came to find that there's a molecule floating around in mammalian bodily fluids and all these different plants that is such a powerful psychedelic, but that just completely flips reality inside out. But at the same time, it's so chemically similar in structure to a major neurotransmitter. So that just blew my mind, that question. And yeah, when I was looking for my graduate studies, it really drove me. And so originally I wanted to maybe work with Steve Barker because of the chemistry background. I thought that was like a perfect fit. But he was sort of winding his career down at that point and wasn't accepting graduate students. So um, yeah, so Rick got me in touch with Jima Borjigan at the University of Michigan, and yeah, she was doing exactly what I wanted to be doing. And that was looking at using analytical chemistry techniques to look at endogenous DMT, using some kind of cutting edge stuff. So the technology, had been established for a while, but cutting edge just in the application. And around that time, and I think it's 2013, the pineal gland paper had come out with Steve Barker and Rick and Gmo, and so that kind of got me pointed in her direction because it was also chemistry, what I was familiar with. Sorry about the emails. um, and so yeah, so Rick got me in touch with Gmo, and I applied to the master's program out there, and was accepted, and ultimately went to pursue the endogenous DMT question there.
So
Uh,
what year did you join Gmo's lab?
2014.
Okay, so the year after they came out with the pineal gland DMT paper.
Yeah, it's 2014.
Okay.
And yeah, I worked on that for the year, and then stayed on for my PhD, and I was co-mentored with Gmo and then George Benshore and Dinesh Pal, and worked on the DMT project to kind of, we had enough like preliminary data at the end of my master's to start to see a pretty interesting thing that ultimately became the 2019 paper in Sci Reports. Uh, but we had to do a whole bunch of different experiments and navigate the peer review process, et cetera. So yeah, so
So
my other mentors were gracious enough to let me work on that in my free time.
so when
Translation weekends and evenings.
yeah, when would you say that you actually started the actual research for that for the data that you presented in 2019? When did that start?
2014.
Wow. So it's 5 years prior. And when would you say that the data was actually finalized before the peer review process took place?
Um, well, that's kind of ongoing, because so I think we originally submitted in 2016, maybe. And then, we went through the peer review, and we were asked to do a battery of different studies. And um, yeah, I mean, so the downside to that was it was, the lab didn't have a ton of funding, and I was working on my main PhD project, which was looking at anesthesia and the role of the frontal cortex and consciousness and how anesthetics work. So yeah, it was hard to kind of balance both of those things and still, do really 100% effort. But that's academia. You have to do 100% effort, split 15 ways. However that math works out.
I mean, it's amazing. I think a lot of people that aren't scientists, they don't realize the amount of work and the amount of time that goes into these studies. They just read them. They assume that maybe they can get banged out in a few months and, you can go on to the next study. But I think especially the importance of the 2019 study, we haven't gotten into the details yet, but people need to know that a lot of blood, sweat, and tears went into that. And it's a very monumental paper. So let's get into that. Actually, before we get into that, let's go a little bit into whatever's going on with the whole DMT, endogenous DMT debate. Because I was watching a few podcasts out there, a few neuroscientists and people in the space talking about that there's actually like there's a debate regarding endogenous DMT. I guess that first started with the whole pineal gland DMT thing. That the paper was published out of Gmo's lab, and then I think there was a critique from a guy named Dave Nichols that stated that there probably wasn't enough DMT produced by the pineal gland to have any physiological effect. And then you went ahead and published your paper that seemed to throw a wrench in his argument. So let's just kind of take it from there. What was the data that was showcased in the paper?
So the 2019 paper, so a couple things. So we already knew from the 2013 paper that there were quantifiable levels of DMT in the pineal gland of rats, of living rats. So for those unfamiliar with the study there, we used a technique called microdialysis. So we were placing a probe in the area of the pineal gland and also part of the visual cortex, which is responsible for what you would think of the visual cortex, vision. And in that study, they were sampling the fluid that would come out of that probe because I wasn't in the lab yet. And what they found using some pretty bread and butter like chemistry techniques, high performance liquid chromatography coupled to what's called mass spectrometry, they were able to show that there was DMT present in the fluid. And that was GMO collaborating with Steve Parker. And so when I landed in the lab in 2014, we started to extend on that finding. So a couple questions. When I joined, that they were doing some immunohistochemistry, so they were looking at like post-mortem brain tissues, looking at protein expression of the genes that, so the proteins that were enzymes that were responsible, or maybe not responsible is the best word, but capable of biosynthesizing DMT, at least in terms of like in vitro, meaning like if you purify these enzymes and you put them in a dish and you feed the DMT precursors to these enzymes, that you can get DMT out. So what we wanted to do was really put the pieces together. And for that study, we said, well, we found DMT in the pineal gland. This probe was also sampling from the visual cortex. And no one has ever shown the co-localization of both of these enzymes. It's either one or the other that people have kind of looked for. So we reasoned that if both of those enzymes are in the same brain area where we're finding DMT, that it would just really increase the odds that DMT was being actively produced there and it wasn't just some metabolite. Because prior to that study, it was a valid argument that maybe this is just some trace metabolite from serotonin metabolism, et cetera, or the breakdown of one of the many other molecules that use dietary tryptophan as a precursor. So we looked in the area of the pineal gland and we found high expression of both of those enzymes, which are INMT and AADC. And those are the acronyms, but we'll just leave it at that. You can Google it.
Yeah, Google that.
Yes. I'm feeling kind of fried. I have a long night ahead of me. But anyways, so yeah, we found co-localization of those two enzymes in the rodent pineal gland. And we reproduced the original finding of DMT in the rodent pineal gland. We also, in the area around the visual cortex where the probe was sampling, found co-localization of those two enzymes in neurons. And so then we said, well, it seems possible that both these brain areas can be producing DMT. So then we also, in a subset of the rats, we removed the pineal gland and just sampled from the visual cortex. So it's like surgically remove the pineal gland. I didn't do that. That was a brilliant surgeon who still works in Dinesh Pal's lab, Taechung Liu. But anyways, so in those rats that the pineal was removed from, we still saw DMT. So suggesting that, well, not suggesting that, showing that the pineal gland at the very least isn't necessary for DMT synthesis or for DMT to be present. Because again, like this is all very, very new research. There hasn't been a lot of labs looking at this, so it is important to be cautious in overextending the findings. Even saying like biosynthesis of DMT, it's like, well, we didn't quite show that. We put the pieces in place that's likely. But then one of the other things we did do is a prior study in Jima's lab had looked at changes in neurotransmission at cardiac arrest using that same protocol with the microdialysis. So sampling the brain fluid in a rat that is being euthanized. And when we looked for DMT in those animals, we saw DMT increases in response to that cardiac arrest or that asphyxia or that stress, if you will. So showing that, there's this physiological event that can cause a change in the level of DMT. So all those pieces together seem to indicate that in the rodent pineal gland and the visual cortex, DMT synthesis is very likely. And then the last thing I think we did that honestly to me was the most revealing and the most shocking, honestly, was we just quantified, we compared the peaks that we were seeing for the DMT to the serotonin. And the way this is done, it's just kind of like a like dissolves like type of thing. There's a column that you pass the brain fluid through, and you also pass like a known amount of whatever molecule you're interested in. And there's more to it than that. You have to collaborate everything. But then the molecules stick based on their polarity to the column. In our case, it was a very non-polar column, or like a fatty column, if you will. And so the molecules will stick based on their dissolvability in whatever the column's packed with. And then when you elute them with a gradient of like ethanol or another solvent, then they come off at different times. And you build these standard curves based on the concentrations of the known amount. And then you could fit the unknown peaks that appear in the same place when they read it out it's called chromatogram. And then you could look at that peak and say, well, that peak's appearing where we see the DMT appearing or the serotonin appearing when we pass it through. And people have done this for a long time with both DMT and with serotonin. But our study was looking at the brain fluid coming right out, as opposed to looking at like peripheral fluids where DMT has been much harder to find, peripheral being like plasma or urine, so it has a chance to be broken down as it's metabolized throughout the body. And when we did that, when we compared the peaks of serotonin to DMT, we saw in the pineal gland, we saw, and in the animals with the pineal gland removed, that serotonin DMT was in the area of the ballpark of serotonin, about half of serotonin. So we were finding serotonin in ranges that have been published on in and around the visual cortex. And a couple nanomolar, and then the DMT was there, and a little bit less than that, about half of that. Suggesting that, hey, like this is in a concentration that is likely serving some sort of physiological function if it's in the ballpark of a major neurotransmitter.
Yeah, so the critique regarding the pineal gland not being able to produce enough to induce some sort of potential physiological, I guess, effect, that had kind of been addressed because you found that DMT was produced at similar levels or half of what serotonin you found, as well as being produced throughout the brain. Is it true that you also found the enzymes INMT and AADC throughout the cortex and the choroid plexus?
Oh, yeah, that's true. We did look in that same study. We looked at a bunch of different tissues, not just those areas. We didn't sample the DMT from these other areas, but DMT has been found in the cerebrospinal fluid, and the choroid plexus is like the site of that synthesis, and it flows through there. And yeah, we saw those enzymes co-localized in there as well. And then interestingly, when we looked in the peripheral tissues, for the most part, there was a separation. So INMT has been found in high concentration in the lungs. And when we looked, we did see it, but we didn't see that co-localization, suggesting that it's a little less likely. I don't think it's impossible, because there's two reactions. There's tryptophan, which is like from Turkey, the dietary tryptophan, which you have to take in. And then tryptophan is converted to tryptamine with the AADC. And then through this like double methylation process, it becomes DMT. So if both those enzymes are there, it's more likely that you can take tryptophan to DMT. But if there's tryptamine around, then it's probably, it's possible. But Yeah, in the peripheral tissues, we didn't really see, we saw segregation or just one enzyme over the other. Aside from in the adrenal gland, there was some co-localization in the adrenal gland, and that's all in that paper. But I mean, I think where the argument, comes from, or the debate, I mean, it's a valid debate because we just don't know yet. But let's have a debate. Let's not just shut it down, right? It does need to be debated because it's one study, and it needs to be our study, and it needs to be reproduced and sort of analyzed in multiple angles, and studies designed to put it to the test, right? But again, I think that, well, not again because I didn't say it yet, but the debate I think really stems from the idea that, can DMT reach a level that would cause hallucinations? And we just have no idea. And I don't know. I mean, to me, I'm really most interested in, and I don't write that idea off, but I'm most interested in just like understanding what it's doing there at the level half serotonin. So I don't necessarily think that it needs to be this like altered reality, naturally occurring mystical states of consciousness molecule. I'm just interested, like, it's sitting in the visual cortex, like, is it involved in visual perception? And maybe that's why when you take this huge exogenous dose of it, you have these like crazy visual, breaks.
Yeah, I mean, I don't think anybody's ever heard you state that DMT is the sole catalyst of altered states or even visual percept, because we all know that there's dozens of neurotransmitters and chemicals in the brain that affect. I think there was that 2015 paper by GMO that showed the brainstorm during following cardiac arrest, where you
Right.
guys saw huge fluctuations and all sorts of things. I guess one of the issues in terms of this guy Nichols was that he was attempting to downplay DMT while at the same time, I guess, upplay, if that's even a word, dynorphin. Um, what are your thoughts on that whole endorphin aspect being the cause of these altered states rather than DMT?
I mean, like, I have all the respect in the world for Dr. Nichols and everything that he's done for the field, first of all. But I mean, I think it's when you think of something like an NDE or something like that, I think the first thing is really, we don't have a lot of experience in Western society with like altered states of consciousness to really like understand a whole spectrum of altered states of consciousness, from psychedelics to NDEs. I mean, I don't personally think that like a psychedelic state and an NDE necessarily have to be the same thing. You know what I mean? They could end up being very, very different things. We just don't really have the bandwidth to understand the differences because, I mean, we know this reality. So I think altered states tend to be like lumped together, and it very well may be that there's different mechanisms at play. Um, and it also very well may be that, you know, something like an NDE is really kind of dictated or facilitated by an individual's pharmacology, right? Because like everybody's different, right? Like, some people have one drink and it's a shit show, right?
Yeah.
And other people can drink quadruple IPAs and
I happen to know some of these crazy
yeah.
people. By the
Right.
way, did you see that recent article regarding that 87-year-old that they got the EEG data on him dying?
I did. Yeah, I did. That fascinating. I mean, the research, yeah, we haven't looked at a lot of this with science. And, I've always been extremely interested in applying the scientific lens to altered states of consciousness. And I mean, it may end up that there's a ton of overlap, right? Because
Yeah.
there are some biomarkers that appear to overlap from what little we do know about near-death experiences. And, we obviously know a lot more from the science perspective about psychedelic experiences.
Yeah.
But there are a couple cool papers out there that have compared like the phenomenology and like the different pharmacology systems that are at play. I think ketamine is like one of the highest ones, right? And in the GMO 2015 study, she did see a lot of glutamatergic changes in the dying brain. So you know what I mean? Like it could be that certain individuals are that have reported in, because I mean, I'm not super familiar with like NDE literature. I mean, I have done in the past, I did a lit review. It never ended up getting published. Like I was working with GMO, just we did look at like hundreds of NDEs, and there were a ton of similarities, but there were some differences too. And you know, it's likely that there's all sorts of different neural compounds at play.
Yeah, for sure. I guess the interesting part in regards to the recent paper was that it seemed to have some sort of overlap with the 2000, was it 2013 or what year was it where you guys published out of the GMO's lab the I guess the gamma surge following the
Oh
cardiac
yeah,
arrest?
the PNAS paper. Yeah, that was my both of my co-mentors, Gmo and George Meshor.
Yeah.
Yeah, yeah, they saw the gamma thing too. It seems
Yeah. So,
to.
I mean, there's a certain consistency. At least it was one just one subject, but at least it points to some sort of overlap with the animal studies because you know that's always a good thing to see. You know, I had a question because one of the other, I guess, arguments that I'd read through Nichols' argument was that serotonin is a hallucinogen. Can you speak a little bit on that? Because in all the things that I've read, I know the 5-HT2A receptor, you know, is obviously correlated with a lot of hallucinatory phenomena, but serotonin itself being a hallucinogen, has that been established?
Um, so I mean, I guess it depends on how you frame it, right? Like things like MDMA will certainly really elevate serotonin, and you can certainly get some hallucinatory headspaces with that.
But at the same time, the same thing with 5-HTP, right?
Um, yeah, I mean, I think so. I'm not as familiar with that, but yeah, I mean, there's right, like they hit the Earwood bolt. Uh, yeah, so I mean, that's like the $1 million question, right? It's like how do all these molecules that kind of tickle the same receptors have these like different effects? Like what's up with you know why does DMT bind to 5-HT2A and serotonin also does, but one of them makes you have this crazy experience. I don't know. I'm not a pharmacologist, but it's
I'm just trying to get a hold of like where these arguments are coming from because in certain cases they seem a little bit discombobulated, and I look for consistency and you know some sort of proof of concept because I get it if you want to debate or you know put a little shade on the DMT endogenous correlating with the NDE thing, but you know at least have your arguments be already proven so you can offer a counter or something because especially for that dynorphin argument because there was a 1999 study showing that dynorphin injected intravenously into human subjects the same amounts that Rick Strassman's study in the 90s. You know, they were producing hallucinatory phenomena with different levels of DMT. And the 1999 dynorphin study was the same levels, and they didn't produce any hallucinations. So I was trying to understand where the whole. I know Nichols cited that dynorphin has a deep affinity for the kappa opioid receptor, but that within itself doesn't prove anything, right? I mean, you need to have the subject saying yes, I hallucinated in order to prove like verify that it does cause what you think it would cause.
yeah. I mean, like I said, I think that NDEs are likely a brainstorm of neurochemistry. Dynorphin may be involved. I don't think we should be closing the book on DMT's potential involvement in that. I mean, I will say that in our studies we saw way higher serotonin increases than DMT. Like it wasn't even close. Even though they were seeing similar at baseline, the serotonin went up much higher. But I mean, it's one brain area. First of all, you know, I would have loved to have stuck around and done and repeated those studies in multiple brain areas because with that microdialysis probe, there's a reason that after every study we verify with histology, like where in the brain it was, because if you move that probe even like fractions of a millimeter, sometimes changes everything that you're seeing. So there's discrete brain areas that have discrete functions. So just because you see one ratio of something in one brain area, doesn't mean that's the way it's going to be. I mean, even serotonin fluctuates too, right? So I mean, even at baseline levels, one could imagine a potential possibility for serotonin to be lower than DMT potentially in a certain brain area. I mean, you look at it in the pineal gland, you look, and it's been well established. Like most of GMO's work has established this, that you see fluctuations in serotonin synthesis, like when melatonin production turns on. So like the brain is, it's not a static machine.
It's not a petri dish, huh?
It's not. Yeah, no, exactly. I mean, there's right. I mean, in vitro studies are very important, but like once you start to get into the brain, like things get way more complicated and the story changes.
Yeah, I hear you. I would like to see that verified if serotonin at really high levels, like maybe you could do an intravenous 5-HTP study and see if you jack up just serotonin levels, whether that induces hallucination. Because I'm a little bit skeptical of that just based on the literature I've read. What do you think about the potential? Do you think it's already established that endogenous DMT or DMT as on its own has neuroprotective effects and anti-inflammatory effects?
Yeah, I would imagine. I mean, yeah, there's a lot out there on things like melatonin, even that it's pretty chemically similar to, that have that effect. And I know you're aware, like the stroke studies that are ongoing, with treating with microdoses of DMT. I'm very interested to see the results of that. Uh, yeah, I think that's definitely one plausible role for endogenous DMT, is like a neuroprotectant.
Yeah, so basically it would be almost like an autonomic method for the body to, I guess, deal with hypoxia and things like that during
Mhm.
the stress states.
And again, like something like that, when you wouldn't even, it doesn't necessitate that it reaches a level to engage 5-HT2A. Um, there's a lot of receptors in the brain that it has an affinity for. So
Yeah, I hear you. What do you think about that whole endogenous hallucinatory agonist system that Barker refers to, in which you administer compounds like LSD? I talked to him and he stated that he wished he had published that study. Unfortunately, his buddy died, but he administered LSD to rats, and then they saw 1,000% increase of 5-MeO DMT and a 400% increase of DMT, both endogenous. So he thinks that LSD might not be a true hallucinogen and rather just pressing the button on our endogenous system. What do you think about that?
I think it's a very intriguing question. Yeah, I think that's something worth looking into. And I mean, again, like to go back to the NDE thing, like there's a reason that these hypotheses exist. I mean, it's because these states do have a lot of similarity, like the reports of NDEs and uh, taking something like DMT. And let's not forget like about 5-MeO DMT, which also appears to be endogenous. There's just way less literature and way less studies on that, which much more mimics the from the what I've read, that mimics the NDE experience that has been reported. So yeah, I mean, to think that maybe some of these compounds that have these hallucinatory properties that are produced endogenously, um, are upregulated by something like LSD or psilocybin. Yeah, it's a really intriguing hypothesis.
We'll see if we can get some funding done to go ahead and carry out a replication study of sorts. Another question, just kind of out of left field. I don't know if you've ever heard of a guy named Sean Thornton. He was an artist. So basically, he would draw paintings and things that kind of mimicked what ayahuasqueros draw on their visions or DMT visions. And
Yeah,
he had
I've used some of that in one of my talks, actually, as like a acknowledgments background.
that's cool, man. Yeah, because I guess we know that what is it called, acromegaly or whatever, in terms of if you have a tumor on your pituitary gland, and that sometimes induces an increase in human growth hormone. And I think Sean Thornton had a tumor on his pineal gland.
Yeah.
And
Yep.
obviously, it's the debate was whether the pineal gland can produce enough DMT to cause an effect. And obviously, we know he's not getting those effects from the tumor squeezing the pineal gland to produce melatonin.
Yeah. There's the publication that shows there's one out there that shows like some visual phenomenon that the person that had the tumor drew.
Mhm.
Like, I don't think it was Sean Thornton, but it was a case study.
Okay.
They looked pretty DMT like. They didn't mention that at all in the publication, but you look at the drawings
Yeah.
and then you look at something like from Chris Timmerman's, because they included some of the artwork, and it's like, Well, it's interesting that you say that, because when in those rats that the pineal gland was removed from, we actually saw an increase in DMT when we sampled, and they had ample time to recover. It wasn't like we just like started sampling like right after the surgery. So yeah, who knows?
Yeah, who knows? But at the same time, it is pretty interesting as an anecdote in terms of here's a man with a pineal gland tumor and he's drawing all these visionary states, and we know that it's not coming from melatonin, and I don't know if it's coming from serotonin, but I would tend to think it's probably DMT, but obviously I'm biased. How does like blood plasma measurements compare to what's going on in the brain? So let's say you see like 1,000% increased spike of something in the brain. Does that automatically correlate to seeing a spike in the plasma, or how does that work?
Yeah, you know what? I'm not like super versed in that literature, honestly. I have looked a little into the like serotonin for that. Um, it seems like it's really dependent on a lot of things. Like it's dependent on kind of like what the molecule does too. Because like for instance, like DMT will cross the blood-brain barrier, but something like serotonin, like most of it's produced in the gut, right? So it really, I think it depends on what you're looking at a lot of the times, and understanding kind of like how other parts of the body, what's going on. It really depends.
I get you. Yeah, it was just another strange argument with this whole debate thing was that you were supposed to see some sort of pharmacological type response of DMT in the periphery during like a near death experience. And that was like an argument as to why, or that was like what would be needed to quantify DMT having any role in the brain, which I found kind of strange because I've seen other studies where something will spike up by 700-800% in the brain, and then in the periphery it's only like 40%. So
There was what compound in the
I
studies?
think it was melatonin.
Oh,
Melatonin.
really?
Yeah, that it was not as the percentage rise in the plasma wasn't as high as in the brain.
Yeah. No, I've done some work with sampling melatonin and uh, like human research. And yeah, it was like melatonin's pretty reliable. Like we were able to even just use, I think we used saliva. Um, but there's the newer study by Ashton, I believe, right? And they have a pretty highly sensitive plasma DMT assay, it seems like. And of note is they saw, if I'm interpreting this right, because I kind of read it quickly, but they did see like one of the main DMT metabolites that was kind of contaminating. So DMT, endogenous DMT itself, they didn't really see any like, well, let me back up. These were data that came from the IV infusion studies that the Imperial College Group was working on. And if I read it right, they didn't really see much endogenous DMT in the periphery that was messing up their quantification of the bolus dose from the IV, but they did see some contamination from endogenous one of the main metabolites of DMT, which is interesting to me, right? So I don't like, maybe in the periphery it's being really broken down at a rate that it's just kind of impossible to get a sample on it, is
Yeah,
kind of what my interpretation of that might be.
but
But
maybe measuring the metabolite from an endogenous perspective might be a viable way
right,
to measure.
yeah.
what's going on in the brain.
But I think DMT is likely produced in the gut too. We did try to throw the antibodies. So back when we were doing protein staining, what ultimately made it into our 2019 paper was the mRNA, because it was just much more stable and less messy. But we did look at like some gut samples and like intestinal tissues and things like that. But there's so much blood, and with immunochemistry, anytime there's like a lot of blood in an area, you get a lot of false positives. But there was David Olson's group published something showing that if you administer an antibiotic, you can basically wipe out. There's a ton of DMT in fecal matter, and for whatever reason, we poop a lot of DMT out, right? Maybe it's just kind of like it has like a peristaltic effect or something, just. But it seems like it's being produced in the gut by bacteria, because when they administer that antibiotic, they like wiped it out, and then they let it come back after the antibiotic, and the DMT levels rose again. So it seems like it's in the gut.
Yeah, maybe 10 years from now or 20 years from now, depending on the funding, we'll start to realize that DMT is kind of like ubiquitous throughout the system, like serotonin and melatonin.
Yeah, I mean it seems like that's the case. It seems like it's around, and I don't know. I mean, our bodies are labs basically, right? Like they're chem labs. They just make a ton of different molecules. So
do you know if
it's in there? I mean, I think it's probably doing something, right? Like, I mean,
yeah, I mean, at least it seems based on the literature that it does have some, I guess, cellular protective properties and anti-inflammatory properties that it's just at its most basic, but
right. But I mean, the fact that it's so similar to serotonin too, it's not out of the question to think it's involved in like processing consciousness in a sense. I mean, what is consciousness, right? It's mediated by your senses for the most part. So like if DMT were involved in processing visual information in some way, which may be the case since it's in the visual cortex of rats, it's in a sense mediating reality.
Yeah. Let me ask you a question. Do you know if there's any compound produced in the human body that's known to have no function whatsoever, that doesn't do anything?
That's a great question. I have no idea, to be honest with you. Yeah, I'm not sure. I mean, we could probably Google it right now together.
Well, I just
I mean, I don't know. Have you come across that?
I talked to Barker and he said no. So I mean, what I'm saying, the only reason why I bring that up is because I've seen that thrown in terms of, I don't think Nichols threw that in, but other random people threw that in as just kind of like a side throw to the DMT debate, which I found kind of silly because I don't think that there's any compound in the body that's known to have no known function. We might not know its function, but having no known function is something else altogether. And if that's the case for DMT, it would be the first, which is kind of, it's like you're going out of your way to be a little silly.
I was gonna say, I mean, like maybe a bunch of metabolites. My biochemistry is a little rusty, but I feel like even if you look at something like the Krebs cycle, that all those metabolites like feed into other things. So if something's around, I think it sort of evolves to have a purpose or it goes away.
Yeah, that's what it seems like, man. Let me see this. I know you've had a long day, so I'm gonna cut this a little shorter, but
Oh, it's all good. I'm in the zone now.
All right, let's keep it going then, man. We'll start talking about all
tea and
start talking about
the Grand will still be there tomorrow.
Start talking about the fights, man. So what's going on with you right now? What are you researching? I know that you're not at University of Michigan. You wanted to stay there because you want to continue the DMT research. You were forced to kind of look other places that can keep your career going. You're at where UC San Diego these days. So what are you studying these days?
Well, I mean, when I was pursuing a postdoc, the big thing was I wanted to learn human neuroimaging, and yeah, I reached out to a couple groups that were really interesting to me, and got the shot out here with Fadal and we're doing some pretty exciting stuff. Like we're working on multiple, so everything's based in understanding chronic pain and understanding treatments for chronic pain. And so when I first kind of started reaching out to people for my postdoc, that really intrigued me because one of my mentors at Michigan, his name's Rick Harris. He also works in the chronic pain research realm, and he was really into meditation, and I took one of his neuroscience and meditation classes, and that's where I found out about Fadal's work, because has done some cool stuff on endogenous opioids in meditation. So I've been fortunate to be able to continue some of that research. So some of the stuff that he's shown over the years in healthy people that have just like been able to pick up meditation, so naive meditators, that as little as like 4 training sessions can overturn this noxious heat stimulus that you apply to the calf. So it just heats the skin up, the surface of the skin, to like 48, 49 degrees Celsius. And
I've been in one of those horrible studies, man.
oh, really?
Yeah, they put that heat pad on your back and they say, let's see what happens to your brain in fMRI. And don't move a half a millimeter.
Right. Yeah, oh, yeah, you gotta be still. So that study showed that when you administer an opioid antagonist, naloxone, that basically the pain reducing effects of meditation hold, right? So meditation isn't mediated by these endogenous opioids. So that was really interesting to me because of like the endogenous DMT, right? And so I got to continue that study out here. We just finished it up actually, and we're starting to look at the data now to write up for publication. We repeated it in individuals with chronic back pain because there's some differences in the endogenous opioid system in individuals with chronic back pain. So we're doing that, and then we have a ton of other studies that we're working on with meditation for chronic pain. So looking at like you said, like brain changes during meditation after inducing pain, either with this thermal stimulus, or we also do something called the straight leg raise, where we'll actually induce some of the lower back pain by raising the leg of the participant. And then we have them meditate in the scanner to see if meditation is if it's possible to reduce the induced pain for their endogenous like chronic back pain instead of just that heat stimulus, that exogenous heat stimulus. And we scan their brain, we're interested in the neural correlates of that analgesia. And then we have some stuff coming up with cannabis for pain, and then also a psilocybin study that'll be launching very soon, and that one's for phantom limb pain. And there are a lot of reports from the 60s. There were some studies back then looking at psychedelics for phantom limb pain, LSD, etc. And it's shown some promise. And then there was a case study recently from a group with an individual that's in our group, showing that psilocybin coupled to mirror therapy, which, if you know what that is, where they'll actually place a mirror sagittal to the missing limb, and just the being able to see the perception of the limb will alleviate some of the pain. So when this individual did the mirror therapy, it helped, and then it kind of went away. And then when he did psilocybin, he did, it was kind of like in the desert, like rogue type of thing, but it was still written up as a case study. He was able to also get that effect, but when he coupled them together is when he got like the biggest effect. So we're starting just with psilocybin and not like introducing the mirror therapy component to it. But it'll be very interesting to see. And I mean, I've already had the pleasure to speak with some people who reached out to us that have phantom limb pain that have tried psychedelics for it, and they're really excited by the study because they they found some serious relief from psychedelics.
That's great. I think people need to realize that you have to start with anecdotes and a lot of science, right? You need to start with observation, and then that's a starting point. It's not an ending point, but we can't just discard things and just call them anecdotes.
You
And
got to
just,
listen to people. Like people's experiences are like that's where it starts. Like,
yep,
I mean, you do have to quantify it if you're approaching it from the science vantage. It needs to be reproducible over and over again and have good placebo controls, etc., whatever you want to call it. Yeah, because the placebo effect's real. But so yeah, I mean, it's important. I mean, I think that, so the late Ed Domino from the University of Michigan, a story that he always told that really resonated. So for those that don't know, Dr. Domino was very instrumental in introducing ketamine into the clinic for anesthesia. And he would get these reports early on, like way before ketamine was established as an antidepressant. I don't remember the exact years, but he was recently on Hamilton Morris's podcast before he died, and he talks about this. But he says that one of his patients that he was seeing was telling him, I've been doing ketamine for depression. And he was like, dude, you can't do that. Like, no way. Like, let me give you, this like first generation, like SSPQRILT, whatever, let me just like fiddle with your noradrenaline and all will be well, right? But he's always like, Jesus Christ, John, if I would have known what I know now, like he's like, you got to listen to like people's anecdotal evidence, like for sure, right?
yeah,
Like that's how you, especially in a patient setting, because it's the whole purpose, right? Like it's, around here we say like, for instance, placebo, right? Like if it works, like it sort of throws a wrench into the whole concept of placebo because it's working, right? So it's like, let's understand how it works. And we
yeah,
do some of that here. We found that meditation is more effective than placebo, and different, because so like placebo does engage opioids for its effect, right? And so you can like reverse some of the effects of like a placebo mediated analgesia by blocking opioids.
wow,
Yeah,
that's
yeah. So, and then the brain mechanisms are slightly different too.
that,
So yeah, it's wild stuff, right? But
yeah.
like you said, listen to these people, and if it works, it works, right? Like, and that's a big thing around here. It's like, we want to know how it works, certainly.
Yeah,
But at the end of the day, if people are getting relief from it, then we've done our job.
speaking of listening to people, I have a story for you. The first time I smoked DMT was actually I had an EEG on, and this was with the scientist Dr. Juan Acosta Urquidy, rest in peace. He published a paper in 2015 regarding DMT and 5-MeO DMT and EEG correlates of smoking it. Anyhow, he fits me up with the cap. He gives me the pipe, and we get to work. And basically, I rip it a few times, and he asked me, "How are you feeling? Are you in?" I'm like, "Nope, I need more." So he goes ahead and fills the pipe again. He's like, "Rip it harder," and that's what I do. And when he's looking at the EEG map, he's seeing basically the classic signal for a transcendental experience from DMT, the alpha drops, and seeing the gamma surge and things like that. But I didn't see anything. I didn't have any visionary experiences. I had deep somatic experiences in terms of disassociation from my body and feeling very numb. But if you were to speak to him, he's like, "You're in, you're in. I know you're in. I know you're having an experience." And then
This
for
was
me,
NN.
that was NN, yeah. But that's what I'm saying. It's interesting. You still have to listen to the people, regardless of the data that you're seeing. It could be very, I guess, consistent in terms of people that are having experiences, but at the same time, it doesn't always mean that, right? So it's like, it's yet you have to listen to the people, regardless of. I mean, the data is important, but so is the person's experience, right?
Yeah, totally, it is. But
Yeah.
you do, the reason that where the quantification steps in is like sometimes in designing a good study, it's like people read stuff, right? So
Yeah.
in this day and age, like if everybody that signs up for a study is like, oh, meditation is going to work for my pain, then they're instantly convinced that that's going to happen, and then which is why like we've dove into that, and so it's so hard to control in a psychedelic study though. I know there's a push to use like um, oral cannabis, like edibles, THC.
Be
And
careful with that, man.
that's what I'm saying. But like, right, seriously, but as a placebo in psychedelic studies, it makes a lot of sense because otherwise you're dealing with things like niacin, and it's like, I mean, which gives you the flush apparently, but yeah, using something that's actually psychoactive, but it's like engaging something different. That's probably the way to go,
Yeah, not to
especially
geek
in
it.
terms of like depression, etc. And honestly, like a high enough edible dose, like you're gonna confuse some people.
But that's what I was talking with Barker about, was the whole heteromer aspect in which two receptors from completely different, I guess, systems can form one, and then you can have like your cannabinoid receptor and a 5-HT2A receptor basically merging into like a coupled receptor, and then you throw a bunch of cannabis on there, and it can stimulate the 5-HT2A activation
I gotta
without.
talk to Barker about all that, man. That's
Yeah, that's
cool.
crazy shid, man. So it's like it just complicates things even further. So when people want
There's
to
a
get,
study out there too, endocannabinoids as monoamine oxidase inhibitors.
yeah,
There's got to be some interplay with cannabis and serotonin, like cannabis for migraine, for instance. Like migraine's definitely has a serotonin component to it.
I'll bet you $20 right now that cannabis edibles upregulate 5-MeO DMT endogenous. Whenever that plays out, we'll figure that out. But $20 on the table right now, man.
Yeah, I suppose it depends on the size of the cookie, right?
Something like that,
Yeah.
man. The whole pain aspect is super interesting because I've interviewed a lady named Sharon Waxridge, and she specializes in hypnoanalgesia and medical hypnosis, where some people are actually allergic to the substance in chemical anesthetics. So the only way that they can actually go through the procedure is either just maybe drinking like 10 quadruple IPAs before, or doing the hypnosis. And she even documents it on her YouTube channel, where there's people that go under hypnosis and boom, they're getting the wisdom teeth popped out.
Dang.
And
Wild.
yeah, I just find that intriguing with the pain studies that you're doing, and maybe in the future we can do some sort of comparison studies between meditation.
I think those are out there. Oh, I see. Yeah, I think that those are. Oh, I mean, that's a space that we're extremely interested in here. Is the overlap between like meditative states, psychedelic states, and especially for pain too. Like, can one be better than the other? Can meditation sort of supplement psychedelic therapy?
Yeah,
If for nothing else, to like ease the headspace, and make it a safer overall experience.
yeah,
Or will it help amplify like pain reduction if indeed it does turn out that like psilocybin is effective for pain. So yeah, a lot of questions. But the hypnosis, there aren't hypnosis meditation. I'm not really familiar with the literature that much, but yeah, there's been some studies comparing the two for sure.
yeah, for sure. So you're the DMT scientist, man. We know you're working on the pain studies and the psilocybin and the phantom limb, but do you still have something like some passion for endogenous DMT research going forward?
Oh yeah. We have some things a brewing just slowly, but yeah, there are some things a brewing that we're excited about. Hopefully this week we'll see some of that data. I've still been working with the Michigan group, and some other folks at some other universities like Minnesota. Um, yeah, so yeah, stay tuned. Hopefully we could translate some of these findings out of the rat.
Intriguing, man. I mean, that's why I keep telling people, there's a large vast of people that are interested in endogenous DMT research. And if we want to see this thing go forward faster and not have to wait 6 years in between published studies, we're going to need to get this thing funded. I know, John, you've been working behind the scenes on projects, but you need that support. So if you want to go ahead and support this man, you can go ahead and donate to DMT Quest, and I'll just forward him the funds, because we can't wait 6 years for studies, man, or else it's just too much speculation, right? In between studies. And you can't blame people for debating. I just ask for like good, clean debate that's based on as deep of facts as possible without throwing out too much nonsense.
Yeah, we need this shit like a Star Wars franchise. We need another publication every like 6 months.
Every 3 months, man. I mean, that would be the thing, right? Like to really just crank out endogenous DMT, endogenous 5-MeO DMT, and even endogenous monoamine oxidation inhibitory studies, right? Like it's there's a lot to be done, and we need to get that thing going. So any thoughts on we're about to get into the strange stuff. Any thoughts into the BICS contest, Bigelow Institute Consciousness Studies? They basically put together a contest last year about the best available evidence of survival of consciousness following bodily death. Were you able to take a look at any of the information regarding the winning essay?
So I haven't dove too deep into that, admittedly, but yeah, it's on my radar. It's a it's a cool thing that they're doing for sure. Yeah, I need to get a little deeper, but yeah, I've checked it out, and it's super intriguing work, and I like that, it's the approach is very from multiple angles, multiple advantages.
Yeah.
I mean, because like, come on, like, is science really going to be how much can science really contribute to that conversation? Honestly, like, it's there's a point where where we analytical stops.
Yeah, I mean, to a certain extent, I think I mean, there's some theorists out there with electromagnetic field theory of consciousness, and maybe that can kind of add some insights as to what happens after we die. But yeah, the Bigelow Institute, they're really going for it in terms of trying to, I wouldn't say prove things, but they're really trying to bring the spotlight to consciousness research and things like perception after death and interacting with that whole thing. So we'll see how far that goes. It's interesting because obviously, you research DMT, so people assume that you're all up into like esoteric sort of thoughts. And what is your thoughts in regards to things like mysticism and spirituality?
Maybe give you a little more specific. I mean,
I guess just let's just like specify the concept of like an out of body experience. So this is a very consistent thing that people describe either in physical trauma or near death experiences where people describe being above their body, seeing themselves down there. Like, do you think that that's purely a construct of the mind, or do you think that there's a potentiality that could be a little bit more complex where it's both biology based and maybe super normal?
I mean, okay, so I think it's biology based in the sense that you do a thought experiment, right? Like, we don't really know what happens when someone dies, obviously. But if the brain were to stop functioning, like, would that experience transcend? I mean, I don't know, man. Like, I the way that I approach it is, for a scientist to be closed-minded to that concept is foolishness, because like, you're not going to prove it, right? So, like, if you can't really touch that with science, how are you going to like shit talk it? You know what I mean? How are you going to be like, oh, well, that's not how that works. It's like you have no idea, you don't have any idea how that works. So, like, you can't really weigh in on it other than to just be like in awe of the fact that people do report this.
Yeah.
I think there's biology that underlies all this stuff, right? There has to be, so let's just talk about like what would like people that have these experiences and they come back, what would the brain look like, right?
Yeah.
And the interest in like psychedelic molecules endogenously, like DMT, 5-MeO, DMT, like being mediators of these types of experiences. I don't know why, like, scientists think that's so far-fetched. Like, we don't have any proof that that's the case at all from a science perspective, and it very well may end up being that's not the case. Uh, but it's a reasonable hypothesis because people have those types of experiences taking these types of molecules exogenously,
Yep.
and then there other people report like similar states that have never even touched a psychedelic. And if you listen to him talk, like I had a friend recently contact me from a really long time ago. I haven't spoken to her in years, and she was telling me about a near-death experience that she had because she knew I was researching DMT, and like the stuff that she was describing sounded incredibly psychedelic. Uh, so to say that endogenous molecules like DMT might be involved, I mean, we don't know. We just don't know, and it is a possibility in my mind. So if that's what you mean about like mystical.
Yep.
Yeah, I think it's completely reasonable. There has to be some sort of biological change. Even if it isn't the chemistry. Maybe it is like some sort of like just overall like shift in how the brain communicates. I mean, I was rooted in chemistry, so I'm interested in the idea that, especially when you take a psychedelic, you see those changes, that it's pharmacologically driven, but that doesn't necessarily have to be the case. We just don't know.
Yeah, for sure. I mean, I want to see more research. Just there's so many systems in play, and it's not just one. They're all interacting pretty in a complex manner with each other. So, we got to be fair to the entire game. One of the interesting studies taking place right now is a guy named Sam Parnia, where he's doing, I think, 500 EEGs on people in hospice, and he
I've
wants
seen
to,
that. Yeah.
yeah, he wants to see what kind of brain changes take place during concepts like terminal lucidity.
Yeah.
What do you think about the concept of terminal lucidity? For those of you that don't know what terminal lucidity is, it usually takes place in an elderly patient in which they're suffering from memory loss, either dementia or Alzheimer's. And then as they near death, they start to become lucid, where before they wouldn't remember their close family members, they wouldn't remember their names, they wouldn't remember anything. And then when they get close to death, then it's like they're almost like themselves again, and they remember everything prior to them dying. So, what do you think about that phenomena?
Yeah, I mean, my PhD mentor, George Mashor, actually wrote an article on that. I'm not sure if he's involved with the study you're talking about. He may be in some fashion, but in some facet. But yeah, I mean, it's reported. Like, yeah, I have some friends that work in hospice, and I'll swap emails with them from time to time and share articles like that, or like psychedelics being integrated into the space of like dying with the option of using psychedelics to kind of ease the anxiety, etc. And some people have said that they have encountered that. It's out there. And right, like there's I would be absolutely shocked if there was no difference in the brain of someone pre and someone during that moment.
Yeah, yeah, absolutely. And I think that kind of there's overlap with things like deathbed visions. Deathbed visions is not exactly like terminal lucidity, but it's when similar like an elderly patient and they become close to death and they start to see people that have already passed away, and they're kind of telling them, don't be scared, you're going to come to the other side, and we're here to kind of guide you along. I mean, some people would say that's pure hallucinatory phenomena. It could be, but it's very consistent and it seems to be helpful. So, that would be something that would be an interesting part of the study as well. Let me see what else we got. I'm just really like fascinated with your journey, the work that you've done so far, the struggles that you've been through, putting out a great paper and still dealing with backlash in the process. But I guess that means you're doing something right, if you got people kind of nitpicking at your work. So that's always a beautiful thing when you can take those hits and keep it moving. I know you've been pretty busy this week, and I really appreciate your time. I think that we need more scientists like you that are determined to go ahead and really just put it all on the table, whether there's funding or not. You need to be driven enough to get the job done. And I think the world owes you like a big thumbs up for all the work you've done and the work that you continue to do. So thank
Nah,
you.
thanks.
Yeah, the
I
humble
appreciate it, man.
The humble scientist, man. Well, we need to get this guy paid. We need to get some dollars, not in his pocket just to drink his quadruple IPAs, but we do need to get his work funded, because this is what I feel to be one of the most important fields to study, to go ahead and tie in some of these mystical states with neurochemistry and electrophysiology, and possibly even other aspects so that science can measure, so we can help kind of quantify some of these states and make people more comfortable talking about them. I think that's an important thing for society. And maybe it could bring people together in the process and make people a little bit more gentle with each other.
All right, brother. Anytime. It's my pleasure. And
All right, Dr. Dean, you're the
appreciate your time
best, man.
too.
Yeah, thanks so much for taking the time to talk with us. And, we'll see you maybe in a few months so we could talk a little bit more about the research that's been going on.
All right, my friend. Have a good night.
Thanks, man.